All right, so today in lecture 15, we pick up basically where we left off last class, talking about Markov models and hidden Markov models for gene finding. Before we start with that though, I'd like to cover a few little things about R, in particular related to iteration. And then, like I said, we'll go to the my notes for hidden Markov models, and then we'll come back to some slides that look at uh, other aspects of gene finding that are used in practice and some of the sort of industrial strength software that's out there. Okay, so iteration, which we've already seen a few examples of it in different formats, different forms, including a few for loops and some apply statements here and there. I wanted to cover it a little bit more carefully here. Okay, so let's start with for loops. They're particularly useful if we know a priori, so in advance, how many times we need to execute some body of code or something, right? So a body of code in R is anything surrounded by two curly braces. In computer science, they really like their braces and they name everything. So you have parentheses, okay, I won't write that out, but that's a parenthesis. And we know that that's very different than a brace or a square bracket. And so we might call that a bracket, or explicitly a square bracket. I would say, and I won't write it all out because my typing's terrible here. And then a curly brace. Sometimes we just say curly brace or curly bracket, right? And they have three different meanings. We know that the parentheses are used for function calls, for example, or for forcing the order of operations. Brackets are used for selecting. We've seen both the single bracket and the double bracket for selecting elements of a list, for example, or columns of a table. And the curly braces are used to denote a body of code. So all the code, in this case here, just one function call to the cat function, belongs to this same body that from the opening to the closing brace. And the for loop here has the following structure. It's ubiquitous. We have a for statement. It's not a function. It's a bit different than a function, so to be a bit careful. And then there's a space here, but then this says that for I in 1 to 10, so we know what this means, right? That this guy here, we've seen this many times now in the course. This is just then a vector 1, 2, 3, dot, 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 10, right? Okay, so I is going to take the value 1, and then it's going to take the value 2, and then 3, then 4, then 5, all the way down to 10, okay? And it does this in this order. So basically this specifies a range of values for the variable called I. All right, and this I is like any other variable essentially. It takes, in this case here, if we go back to our canonical model of what happens in R, it creates a box, and at the beginning it puts 1 in there, and then it's going to put 2, then 3, etc. So how it does that? So it comes in here, and firstly I will set be equal to 1, okay? And now it'll execute this body of code, which is just the function cat, right, to send to the screen. Good job, Aki, you've lifted it 1 time, okay? Oh, sorry, this is in response to the question, how many reps of a 20 kilogram weight can Aki bench press? And so we're going to count them through. So he's lifted it 1 time because I is equal to 1. And then it comes to this closing parenthesis, and now it goes back to the opening parenthesis. But it updates I, and so now I will be equal to 2. And now it executes this body again. In this case here, I is going to be equal to 2. And so it prints out 2 times, and it hits the bottom, goes back to the top, prints out 3 times, all the way down to the last value for I, which is 10. It comes into the body of code, executes that, and then it stops. And then this piece of code down here will execute after this for loop has finished, okay? So we knew a priori that Aki wouldn't be able to bench press the weight more than 10 times, and so we could count with him 1, 2, 3, 4 all the way to 10. Okay. Another example. Okay, so sorry. In general, for loops have this structure. Whoops, and I missed a parenthesis there. For, and then the variable name. In our last example, that was I, in, and then a set of objects. In the previous example, we saw that the set of object was the integers 1 to 10, but it could be anything. It could be names or logicals. It could be basically any object that we would like, and that's what this variable I, or in our case, that example was I, whatever our variable name will be, will take on that set of objects in that order, and each time it'll execute this body from the beginning to the end and redo that. Okay, so yeah, I guess like traditionally for loops in R were discouraged because it slowed things down, just the nature of the language, but that's been fixed for a while now, and there's absolutely nothing wrong with for loops, and I find them that they're very intuitive, but other people don't, so some people get a little bit confused by them. So here's a second example. Now I've set a vector of characters, character vector, right, of all boy bands that I could think of, and then for, here's our for structure, and this is our body, open, and our close is down here, and inside of it we actually only have one statement, which is an if. Okay, so in this case here, if you detect in the bth element of bands the word Justin, it executes this statement, otherwise it executes this statement. So this is an if else statement, which we've seen last class, right? So now we're making a conditional. If the boy band starts with it has the string Justin in it, it will do one thing. If it doesn't, it does something else. But let's take a look back here. Bands is a character vector. So if I want to access, like, say, the third boy band, it would be this guy here, and this would be the third element of bands, right? So now what's happening here is that B ranges from 1 to the length of bands. So the length is going to be 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. So B is going to start at 1, and it's going to go up to the length of bands, which is 14. So here, the string detect is on the bth element of bands. So the beginning, that's the first. And so the first guy is Jackson 5. And when it comes in here, the string detect will be, let me clean this up a little bit, okay? The string detect will come in, and it'll fail because Jackson 5 does not contain Justin. And so it'll come down here to the else statement and execute this. So now we print out on a scale from 1 to 100, Aki rates the bth band, which is going to be the Jackson 5. So that's the output right here. And how much does he like that between a scale of 1 and 100? Well, we use the random function, the random number function. So I choose a random number between 95 and 100 because he loves all these bands, and that's the sample function. And you can see here that Aki loves Jackson 5, or rates them as a 98 out of 100. Then we go back to the beginning of the loop. Maybe I have to be in. Oh, I see. Okay, I got the hang. No, it just took 16 lectures or so to figure out this pizza thing, but I got it. So now it goes back to the beginning, which is up here, and now B becomes the second element in bands, so that's going to be, scroll off here, New Kids on the Block. It doesn't contain Justin, so now it comes in and executes this guy down here again, and again now it selects a number, random number, and it turns out that Aki rates New Kids on the Block a 96 out of 100, so not quite as good as the Jackson 5. And then it goes back to the top. Now eventually it will get to number 1, 2, 3, number 4. B will be equal to 4 down here, okay? And the string detect actually determines that the fourth band does contain Justin, and so it executes this, and Aki rates Justin Bieber to be infinite. Okay, infinite like, maybe love. And it will go through the rest of the list exactly like that and execute each one. And so we'll see something, all of Aki's favorite boy bands down here like that. Okay. Okay. The way I had done this from 1 to length in the previous, I set the range of B to be 1 to the length of bands. And that can be a little bit dangerous. It's better to use this function called seek along, okay? And it's a little bit safer. Okay, so, well, this is, you can ignore this. It doesn't actually work in my slides. It should work in your session. If you want to change your output text, different colors, you can use this crayon package. It's really not really important here. So here, Aki's pumpkin carving success is going to be the empty set, right? And you can witness that down below with this picture. And now, but the thing is, if I ask for, let's suppose I wanted to go and create a for loop, and I wanted to say every time that Aki successfully carved a pumpkin with some skill, I'd say, wow, Aki, that's really interesting. But he doesn't have any successes, so this shouldn't print out anything. However, if I use this 1, if I use P to be 1 to the length of pumpkin carving successes, I get some strange behavior, because 1, that's this guy here, go to 0 is the length of pumpkin carving successes, because he clearly has no successful pumpkins carved. But still, when I run this, it executes this twice. It first prints out, wow, Aki, that's really interesting, and again, so P takes on the value 1, and then it takes on the value 0, which isn't really what we wanted, right? The thing is that you can count down. So R tries its best to understand that. It says, oh, you're counting down from 1 to 0. That's not what we wanted. We wanted to basically say, if there are any successful examples of Aki successfully carving a pumpkin, then we'll go through the for loop. But since there's not, we don't want it. Now, I can rectify that situation by using seek along. Here, again, Aki has no skill carving pumpkins. If I ask for seek along, I get back now just integer 0. So if I say for P in seek along of pumpkin carving successes, now there's nothing there, and there is no output at all. And this makes a lot more sense. It corresponds to our notion of reality, right? Okay. Now, in general, there are other ways, several other ways to implement iteration in R. The for loops are part of the base package, and we'll come back and I'll show you while loops in a little bit. They're also part of the base package. But, well, per is a package within the tidyverse. It has lots of nice functions for iteration that make things a lot easier. Here, I'm just loading the Tara data set, our Tara Oceans data set, and I'm going to select all the columns that begin with bacteria. So that's all the species, all those columns to the past salinity, oxygen, etc. Okay, that's just a straightforward select. I just use the starts with function to select those columns that are bacteria, and then I'll pump my Tara small, Tara S, into this function called map. In this case here, it's map double. Double is basically a synonym for numeric. There's different double functions, one for logical, one for double, one for characters, etc. Here, if you know your data is a double or a numeric, it's the right one to use. And so what it does, when you pass it, when you pipe this tibble into map double, is it sums every column. Okay, which you've seen different ways of doing that now, but here I can sum each column, and I applied the sum. Now here, sum takes this other parameter called NA remove, and you need that to be true because there's lots of NAs in this data. That's map double basically will pass this part on to the sum function. It's like it's called you'll hear this term sometimes called the triple dot, and that means basically whatever else you pass in the map double gets passed as arguments to sum. Okay, and then I took the output of that, which is going to be the column sum for every bacteria that we've detected, unnamed. You can figure out what this. Remember, the names are very long. They're like this bacteria, proteobacteria, alpha proteobacteria, etc. It was kind of ugly, so I put the unnamed in there to get rid of the names on those values. And now this is the first taxon that we identified. That's the total count across all different sites. So that was 102,000 or so. And I printed, that's what this is here, the printout of all the different taxa that were identified. And then I pass it to the summary function, which is kind of a nice little function. I'll scroll down here to the end. It just basically gives back some simple statistics, summary statistics about those counts, saying that the average taxon was had a count of 362. The biggest one, as we know, was 102,000, and there was many with just a single count over all sites, just detected once. And this is your first quartile and your third quartile, right? Think of that as being, if the mean is the 50th, the second quartile, right in the middle. The first and third are between the min and the mean, etc. And you actually see here that there's a big difference between the mean and the median. Most taxa have very few reads that identify them. Okay, that's neither here nor there. The main thing here, I just want to go back to make sure you understand that the map function, I could have written a for loop, and that would be an excellent exam question to ask you, can you make a for loop, use a for loop here instead of the map double, and it would be pretty easy to do. I'll leave that to you. It's a good take-home exercise. Maybe you want to make a note of that right now. Okay, try it out. But that map function basically says, okay, for each column in the tibble, use the sum. Here's another example using a map logical. Here, I've just done something. It's quite simple, but I pipe in my tara small into the map logical function. And this kind of strange notation, this formula notation again. Just think of it as a function, okay? And so my function here is that I sum up each column of tara. That's the notation .x. Map logical takes data, that's .x, and the formula or function, that's .f. Here I'm explicitly saying pass tara s into this sum and with the NA remove, and it'll return true if the number of counts is greater than 50,000. Otherwise, it returns false. And so here you can see that it's only the first 5 taxa that have more than 50,000 reads. In fact, I didn't even know this, but they had sorted the data by, the columns are sorted by most to least observed. So you see these guys here are the ones above 50,000, and then it drops off below 50,000. And this is sorted. You can check that out. Okay, so that's the map logical function, and it works, again, very, very similar to the map double. And instead of a for loop that goes over each column, you could just, you don't need to specify where you begin and where you end. So you can see, like, perhaps in this example it's a bit clearer. You don't need to know how many columns there is. You don't need this variable like I in 1 to n call of tara s. It's all taken care of for you. It just figures out how many columns there are and iterates over them, each one. All right, and then the last concept that I want to talk about today for this little R session is the idea of iteration over of undetermined length. And so sometimes we don't know a priori how many times we need to loop over a body of code. So sometimes it's just hard to predict how many times you're going to need to do it, So in this example, this is just a simple example. We start with a string, a nucleic acid sequence, okay, like that. And I'm going to trim from the 5' end until I get total GC content of over 80%, at least 80% at least. Okay, so here I've written a small little function. This function takes a string, an S, and it returns the string count, so the number of times that G or C appear in S. So remember, this is a regular expression that we talked about a little bit here. So this means either it's a G or a C, okay, and in the string S, okay, that's passed to the function, and I'll divide that by, it's funny, I didn't think my thing was on. Okay, I'll divide that by the entire length of the string times 100. So that's the percent GC, because the string count is, this is the number of G's and C's, and this is the total, including A's and T's, and then I multiply that by 100. So that's just the percentage. And now I write the while function like this. Okay, I have this special command while, and then I have, now this is a condition. It's a logical expression. Okay, that's a logical expression, and that logical expression, of course, evaluates to either true or it evaluates to false, like all logical expressions do. And so here, if this evaluates to true, okay, then I execute my body of code that's determined by this opening and closing curly brace. If the logical expression is false, I stop. So here, in other words, I'm saying, as I modify this string by cutting off nucleotides from the X prime one by one, as soon as the resultant, the resultant GC content is at least 80%, then I'm going to quit. Okay, so here you can see this is the initial string. It starts off with ACTTAGCA, and it comes in and it says, okay, well, calculate the percent GC of all of X. I don't know what that is, but you can see that it's not less than 80%, because here it cut off the first, that string sub, it only takes from 2 to the length of X, so it cuts off this first nucleotide, and it prints out the resultant string. So now it's just CTT, right? CTT. Now it goes back to the beginning, okay, and it checks this condition again. And obviously it's not below above 80%, because it comes in and cuts off this C, all right? And now we're at TTT, right? And it's going to keep going back, and it's going to iterate through this loop, this body of code, each time removing one character, until eventually I get down to here at the very end, where you can see that there are just 7 nucleotides, one of which is A, all right? And it now you know that 7 out of 8 is greater than 80%, so we stop, right? So there, I had no way to knowing really beforehand how many nucleotides I'd have to cleave off before I could stop, and so that's why I used here the while loop. All right, the other thing I wanted to talk about today was gene finding approaches in software that go beyond the hidden Markov model that we've looked at. So, well, firstly, there's a division between eukaryotic and prokaryotic, and we'll talk a little bit about why, but it's generally speaking, gene finding is much different in those two broad categories. So eukaryotic with cells, these are cells with the nucleus, and prokaryote don't. The second, well, I can show you here some of the gene finding software that's out here. This is from Wikipedia. It's not a bad list, actually. You'll see if you go through this that firstly, you'll notice that roughly speaking, you're either a eukaryotic or a prokaryotic gene finder. You're not both. There's some exceptions, GeneMark, for example. If you go through here, you'll see there's a lot that are, in fact, based on hidden Markov models, BGF. Some of these other ones are too, but FGenish. Where else are we here? Come down here to, for example, I've lost it now, I'm sorry. Some of these are also HMM based. Certainly Veil, okay. And a number of other techniques are also prevalent here. Neural net based methods. So we're going to look at some neural nets near the end of the course. Decision trees, support vector machines, or short for SVMs, etc. So there are different techniques out there. A lot of these are machine learning techniques, though, which means that you fundamentally you have a learning set of positive genes and negatives, non-coding areas, and you use that to train a model. It's just that the model changes. Instead of a hidden Markov model, you use something else. Okay. So yeah, and then there's a broad difference between intrinsic and extrinsic approaches. Now we've seen an intrinsic approach already. HMMs are one type. And I should say that they're not just for protein encoding genes. You could use them to, say, detect long non-encoding RNAs. Again, it just comes down to your learning set. You're going to reparameterize your hidden Markov model. The transition probabilities between states and the emission probabilities would have to be readjusted for whatever domain you're working in. But of course, you need the learning set of you need examples of long non-coding RNAs versus non-coding RNAs. Well, that's not quite. I don't need to say non as in actual coding RNAs, but at least you need to. You need to know regions of the genome that aren't non-coding RNAs, and it's all based on essentially machine learning. You have that set of positive and negative examples, and you train your model. Now, I mean, fundamentally, Eukaryote are more complex than Prokaryote, and in a way, then there's more hints in the DNA in Eukaryote for where a gene might be, but it's challenging because the genomes are so big. And for example, in vertebrate genomes, you're only looking at 2% of the genome coding for a gene. So there's 98% of the genome is just random or blank. That's debatable, but people call it junk DNA. I'm not sure it's 98%, but it's a large fraction. And so you think about in a bacteria, your promoter is really tightly constrained around the transcription start site. And so all those short binding sites, for example, and they would have sequence properties, right, that you might be able to train an HMM to find. Those things might be compressed in bacteria and therefore easier to find. But in Eukaryote, these might stretch thousands of base pairs upstream of the gene, and enhancers and these kinds of things might be even 100,000 base pairs, like huge tracks upstream of the gene. So that means those little signals are going to be spread over a much larger area, and statistically that'll be harder to identify. But Eukaryote, for example, have intron-exon structure. Okay, so I've blown it up down here, and you have splice sites, donor and acceptor sites that basically indicate where the splice will occur. You have alternative transcripts, alternative splicing forming different transcripts, etc. So, in fact, these kinds of properties, like the start codon, stop codons for the exons, and these 5' and 3' splice sites, you can use either HMMs or position weight matrices to characterize. So these models for Eukaryote become, in some sense, hierarchical, that you have really an ensemble of HMMs or classifiers in general trying to pick up on different components of what it means to be a gene in Eukaryote. Whereas bacteria, it's a lot simpler. Even the region upstream of genes is a bit simpler, and you have operon structure, etc. So, and the other major thing is that the genomes are relatively small, and there's very little energetic regions, not to mention no introns. And so, it's not really very hard in a way in prokaryotes to find generally where the gene is. You just want to sort of localize exactly where it starts and stops. And that's, of course, not the case in eukaryotes like human, for example, vertebrates, the higher order eukaryotes. This concept of an open reading frame comes up a lot. Genes are most simply identified by the presence of long open reading frames, and that just means that it's really a simple definition where it's just saying either I see a start codon in bacteria, it'd be like ATG, GTG, TGG, etc., or and a stop codon that that should be TAA, THE, or TGA. For example, here in this DNA logo, these are the start codons in E. coli. So you can see that the second and third positions are TG, but A, G, and T are all present in the first position. So there's a bit of a degeneracy between eukaryotes and prokaryotes in that way. That's a very short motif, right? Now, let's go back to, we had this talk in class, in the discussion at least, about being at the Montreal, Bell Center, and everybody guessing, what was it, a nucleotide sequence of like 6 or 7, I think it was. And, already I think it was expected that about every 2 games somebody would win that game, right? But here, it's only 3 nucleotides long, so it's 4 times 4, 16 times 4, 64 possibilities. So basically, all things being equal, you might expect to see an ATG every 64 base pairs as you walk along a bacterial genome. And, well, that's bad news in a way, because I'm not sure it's bad news, but just by random, if you have a genome that's, let's say, 1 million base pairs long, 1 million by 64, that's what, about 10,000 occurrences, I guess. Slightly more than that, I guess. Maybe closer to 20,000 occurrences of ATG just by random chance alone. Okay. So I think glimmer, that was the first one that I knew about. It's one of the most famous that I know for prokaryotes. It's based on hidden Markov models. It's supported by the NCBI. So if you were to go out there and sequence a bacteria, or prokaryote in general, you can go to the NCBI, which we all know about, we're all familiar with, and you can copy or paste your sequence, your FASTA file right there into the glimmer window, and a little bit later you'll get back this kind of response that says, the first open reading frame is from bases 59 to 583, and it'll give you back everywhere it thinks that there is a gene in that genome. Okay, I mentioned previously that there's this difference between intrinsic and extrinsic approaches, and really there's a third technique that's more popular these days based on next generation sequencing, expression approaches. All 3 can be used in both prokaryotic and eukaryotic settings, really, so all 3 really. I shouldn't have said both. Okay, so I don't like these terms, right? I don't think that they're very good, and I kind of think maybe I created them somehow for this lecture, but regardless, let me try to explain what I mean here. Hidden Markov models are an intrinsic method because they take known examples of genes and non-coding regions and they summarize them. In our example in class, we just summarized them basically by their nucleotide frequencies, right? It's just basically saying, what's the frequency of A in a non-coding region, and is it different than the frequency of A in a coding region, right? So it does actually use information from outside sources. It's not completely intrinsic, but it does bring in information from outside, but it just it doesn't bring the whole gene. It's just a summary of the gene. Extrinsic methods, on the other hand, are things like sequence alignment that we're going to start to look at for the next 2 lectures, where you take a known gene, for example, its nucleic acid sequence, and you align it against your target genome where you want to find genes. So the idea is that, if you were to use something like a ribosomal subunit, most likely, I guess it would be just about impossible for the target organism not to have a ribosomal subunit. So what you're doing, it won't be exactly the same, but it'll be highly similar. And so this notion of alignment in computational biology is extremely important. One example is BLAST-X, which is a part of the BLAST family, and it basically will take your gene that you're looking for and try to align it across the entire genome and find where, if anywhere, it aligns well. So these are really kind of complementary approaches. So extrinsic methods are really powerful for finding genes that we already know about, right? So we all expect that we're going to have ribosomal subunits. We're all going to have a proteasome. I think it's probably some of these things are ubiquitous. Not everybody's going to have a nucleus, but if you're in the eukaryotes, everybody's going to have a nucleus by definition. So you're going to have things like membrane transport, and you're going to have chaperones, etc. So a lot of genes are ubiquitous across the tree of life, or nearly ubiquitous, right? So you can use our known examples of those genes to look for similar ones in the organisms. That's really powerful. But of course, if you have a really novel organism from the bottom of the ocean that hasn't been studied, from an ecosystem that hasn't been studied very well, where there might be lots of novel organisms, there may be genes in there that look nothing like what we've seen before. And those ones won't get picked up by these methods, right? Well, there might be domains that they share, but sometimes they'll be too faint. There'll be the evolutionary divergence may be too far, etc. And you won't get them through these extrinsic methods. Now, intrinsic methods will find those kinds of, in principle, could find those rare, interesting variants, but it doesn't have a lot to go on. It's going on just these metastructure of a gene, like how is the gene started, how is it stopped, what sort of structure does it have in between in terms of introns and exons, is there a promoter, etc., can we detect binding sites? All those pieces of information go towards predicting whether a locus on a genome is really a gene. So it has the chance to identify novel genes as long as they carry that sort of that intrinsic structure of what it means to be a gene. A third method is RNA sequencing. I won't go into this, but it's pretty intuitive, I think. If you sequence the transcriptome of a cell, of a population of cells, what you're doing is you're measuring, most likely, it's the sequencer has actually sequenced cDNA that's derived from the mRNA, and that mRNA is derived from the genomic DNA of the organism, right, from those cells. So by aligning your transcripts that you've sequenced back to the genome in a smart way, you can find those parts of the genome that are expressed. And that really has has benefits above and beyond both the intrinsic and extrinsic methods, because it's really almost less of a prediction, right? It's really kind of direct evidence that that part of the genome is expressed. Now, there's also sorts of complicating caveats and special situations there, but it's turning out to be a very powerful method. It makes sense, right? Well, really, when you go to industrial strength gene finders that are supported by the NCBI and JGI for microbes, etc., the Broad, places that are doing a lot of sequencing, you'll find ensembles of techniques pushed together and mixtures of intrinsic and extrinsic methods. This is, for example, how you might do it in eukaryotes, where you would use homology search from databases like the NCBI RefSec to align against the genome to get an idea of where things are. And then you would use bioinformatic HMM-based predictors to predict exon, intron structures, compare them with RNA-seq to look at the transcripts, etc., and cycle through here until you somehow stabilize on what you think are the true genes in the genome, right? And that's these are some of the most famous, most powerful ones currently for doing that. HMMs and R, really, I don't have experience with this DepMix S4. I think it's pretty famous. Of course, hidden Markov models get used in domains outside of biology. I think Seek HMM is for social networking or something, like not like Facebook, but social data. I'm not entirely sure. I've never used those two techniques. I'm sure they're very good packages. I know more Hummer, which is the bioinformatics computational biology software. There is a R package for it, so I think that it's pretty easy to set up hidden Markov models really quickly. And I mean, right now you guys have all the capacity to do that yourself. You have all the components. You would just need to look at the manual and try to massage your data into one of those frameworks. Okay, so points of reflection to end here. Think a little bit about what the relative advantages and disadvantages of these different methods are. Include when you're in your thinking about RNA-seq and how that could be used. Make sure you understand the basic idea there. If you don't, we can talk about it in the lecture. Okay, so next class we're going to start to look at, we're going to carry forward with the Markov models, but towards this sort of in line with these extrinsic approaches of sequence alignment and modeling evolution, particularly in cancer. I'll see you then.